Hello there, and welcome to this Sleepy Science channel. I'm so glad you found your way to this video because tonight we'll be exploring one of nature's most quietly remarkable kingdoms, the world of mushrooms. These are organisms that live between categories, neither plant nor animal, but something entirely their own. They appear overnight as if summoned from nothing, yet their hidden networks may have been growing beneath your feet for centuries. Mushrooms glow softly in the darkness of forests, break down mountains into soil grain by grain, and weave invisible threads that connect entire woodlands. They have shaped the history of medicine, nourished civilizations, and may one day help us heal the planet itself. Tonight, we'll drift through the gentle mysteries of fungi, from the microscopic threads that stitch together forest floors to the ancient partnerships that made life on land possible. Some of what you hear may surprise you, while other truths will settle softly in your thoughts like spores drifting on a quiet breeze. Now, wherever you are, allow yourself to settle. Dim the lights if you can. Let your body sink into comfort, supported by the same earth where mushrooms rise and fall in their patient rhythms. Take a slow breath. There's nothing you need to remember tonight, nothing you need to hold on to. Simply let these stories of fungi wash over you like rain through leaves, gentle and unhurried. Let's begin this quiet journey together. Mushrooms are more closely related to animals than to plants. It surprises most people to learn that fungi, despite growing quietly from soil like flowers, share a more recent common ancestor with us than they do with trees. At the genetic level, fungi breathe in oxygen and exhale carbon dioxide just as we do. Their cell walls are made not of cellulose like plants, but of chitin, the same tough material that forms the shells of insects and crustaceans. This unexpected kinship places fungi in a separate kingdom of life, distinct from both plants and animals yet sharing characteristics with both. When scientists first began studying the genetic code of mushrooms in the late 20th century, they discovered that fungi and animals diverge from a common ancestor roughly 1 billion years ago. Plants had already split off much earlier, taking a different evolutionary path entirely. The implications of this relationship are fascinating. Like animals, fungi cannot produce their own food through photosynthesis. They must consume organic matter, breaking it down with enzymes and absorbing the nutrients. This makes them more similar to us in their basic metabolism than to the green plants that surround them in the forest. Where plants capture sunlight and build sugars from air and water, fungi must find their nourishment in what already exists, recycling the world's organic matter back into forms that can nourish new growth. The chitin in fungal cell walls is particularly telling. This is the same molecule that gives structure to insect exoskeletons, crab shells, and the protective coverings of many marine creatures. When you touch the firm surface of a mushroom cap, you are feeling a material more closely related to a beetle's shell than to the cellulose fibers of a tree trunk. This shared chemistry hints at deep evolutionary connections, ancient branches in the tree of life that link seemingly unrelated organisms. Beyond structure and metabolism, fungi share other traits with animals. Both store energy as glycogen rather than starch. both produce certain enzymes and proteins that are remarkably similar at the molecular level. Even some of the signaling pathways fungi use to respond to their environment, ecosystems found in animal cells. These parallels have made fungi valuable subjects for medical research, as discoveries about fungal biology sometimes translate surprisingly well to understanding human cells. Yet fungi are not animals. They lack mobility, nervous systems, and the complex organ structures we associate with animal life. They exist in a middle space, drawing from both kingdoms yet belonging fully to neither. This is part of what makes them so intriguing. When you see a mushroom sprouting from a fallen log, you're looking at something that defies simple categorization, an organism that reminds us how diverse and unexpected life strategies can be. In the forest, this relationship becomes even more apparent. Fungi interact with plants through symbiotic partnerships, yet their fundamental biology aligns them closer to the animals that walk among the trees. They are decomposers and recyclers, breaking down what dies so that new life can emerge. In doing so, they perform work essential to every ecosystem on Earth, bridging the gap between kingdoms, connecting death to renewal in an endless cycle. To understand that mushrooms are our distant cousins is to see the forest with new eyes. The kingdom of fungi is not separate from us, but woven into the same ancient tapestry of life. Every mushroom you encounter carries within it echoes of shared evolutionary history. A reminder that all living things, however different they may appear, are bound together by threads that stretch back through unimaginable spans of time. Imagine walking at night through a forest where tiny lanterns are scattered across the ground, mushrooms glowing softly with green or blue light, like scattered stars brought down to earth. This gentle radiance is called bioluminescence, the same kind of light that fireflies and some deep-sea creatures produce. More than 70 known species of mushrooms can shine this way, creating their own quiet illumination in the darkness. The glow comes from a chemical reaction within the fungal tissue. An enzyme called luciferase acts on a molecule called luciferin, producing light without heat. This is the same family of reactions that allows fireflies to flash their signals on summer evenings and deep-sea fish to navigate the lightless depths of the ocean. That mushrooms independently evolved, this ability shows how useful bioluminescence can be as a survival strategy. Scientists believe the glow serves multiple purposes. It may attract insects at night, luring them to land on the mushroom and inadvertently carry spores to new locations. The light might also deter animals from eating the fungi, signaling that the mushroom is toxic, or simply making it more visible and thus easier to avoid. Some researchers suggest the glow could even have antimicrobial properties, protecting the fungus from bacteria or other microorganisms that might harm it. The most famous bioluminescent species include the jack-o'-lantern mushroom and the ghost fungus, both of which produce a steady greenish glow visible to the naked eye in complete darkness. The honey mushroom, widespread across temperate forests, can also emit light, though more faintly. In tropical regions, certain species create displays bright enough to read by if you gather enough of them together. their combined glow casting an ethereal radiance through the humid night air. To witness bioluminescent mushrooms in the wild is to experience something that feels almost otherworldly. The light is soft and steady, not flickering like a flame but constant like starlight. It transforms the forest floor into something magical, revealing shapes and textures that daylight or flashlight would show differently. The glow seems to emanate from within the mushroom itself, as though each one contains a tiny lamp powered by chemistry rather than electricity. Historically, this phenomenon inspired countless myths and legends. Before people understood bioluminescence, glowing fungi were attributed to spirits, fairies, or supernatural forces. Travellers spoke of fairy fire or foxfire, mysterious lights that appeared on rotting logs and stumps in the deep woods. These glowing patches were sometimes used as primitive lanterns, with people carrying pieces of luminous wood to light their way through darkness. The glow typically appears strongest in the mushroom's gills or on its surface, where spores are produced and released. This supports the theory that the light helps with spore dispersal, making the mushroom more visible to insects that might visit during the night. The brightness can vary depending on temperature, moisture, and the mushroom stage of development, with some fungi glowing more intensely just before they release their spores. Modern research continues to explore the mechanisms behind fungal bioluminescence. Scientists have isolated the genes responsible for producing the light and are studying how the reaction is regulated. Some have even transferred these genes into other organisms, creating glowing plants and bacteria in laboratory settings. Understanding how mushrooms produce light so efficiently could inspire new technologies for sustainable lighting or biological sensors. There is something deeply peaceful about the idea of mushrooms glowing quietly in the night, creating their own light without fanfare or noise. They ask nothing of the observer, simply continuing their ancient work whether witnessed or not. To know that this gentle luminescence exists, hidden in forests around the world, adds a layer of wonder to the darkness. Even when we cannot see it, the light is there, steady and patient, a reminder that beauty often thrives in the quietest corners of the natural world. Beneath your feet, hidden in soil and leaf litter, there may be a living web so vast it stretches for miles in every direction. Mushrooms are only the visible tips, the fruiting bodies of a much larger organism called mycelium. These thread-like filaments weave through earth, wrapping around roots and stones, linking trees together in an underground network that scientists sometimes call the Wood Wide Web. Mycelium is composed of hyphae, individual threads so fine they are barely visible to the naked eye. Each thread is only a few micrometers in diameter, thinner than a human hair, yet incredibly strong and resilient. As these hyphae grow and branch, they form a network of staggering complexity. A single gram of soil can contain several kilometers of these fungal threads, woven together in an intricate three-dimensional lattice. The network grows by extending its tips through the soil, following chemical gradients toward food sources or away from unfavorable conditions. Where two compatible strains of mycelium meet, they can fuse together, creating an even larger interconnected organism. Over time, This process can produce fungal networks that span entire hillsides or valleys, with individual mycelia potentially covering hundreds of acres. These networks are not passive structures. They actively transport nutrients, water, and chemical signals across considerable distances. When one part of the network finds a rich source of food, such as a fallen log or a deposit of organic matter, it can mobilize resources from other areas to exploit that opportunity. Conversely, when one region faces stress or nutrient scarcity, the network can redirect resources to support it, maintaining the health of the overall system. The mycelium also forms partnerships with plant roots, creating structures called mycorrhizae. In these relationships, the fungal threads extend the effective reach of a plant's root system, accessing water and nutrients from soil that roots alone could never penetrate. In exchange, the plant provides the fungus with sugars produced through photosynthesis. This exchange benefits both partners and has been crucial to plant success on land for hundreds of millions of years. Through these mycorrhizal connections, fungi can link multiple plants together. A forest becomes not a collection of isolated trees, but an interconnected community where resources flow between individuals. A large, well-established tree with access to sunlight can share sugars with smaller seedlings growing in shade. Trees of different species can exchange nutrients through the fungal network. Even dying trees can transfer their remaining resources to younger neighbors, ensuring that the investment of their lifetime supports the next generation. Scientists have discovered that these networks also facilitate communication. When a plant is attacked by insects or infected by disease, it can release chemical signals into the soil. The mycelium detects these signals and transmits them to neighboring plants, which then activate their own defenses in preparation for potential threats. This early warning system operates entirely underground, invisible to casual observation yet vital to forest health. The scale of these networks is difficult to comprehend. What appears to be dozens or hundreds of individual mushrooms fruiting across a landscape may actually be manifestations of a single organism, its main body hidden beneath the surface. The mycelium can persist for years, decades, or even centuries, producing fruiting bodies only when conditions are favourable. Between these visible appearances, the network continues its work, breaking down organic matter, forming partnerships and maintaining the intricate connections that sustain the ecosystem above. To walk across a forest floor is to walk across a living web of staggering complexity. Every step covers thousands of kilometers of fungal threads, all working in concert to recycle nutrients, support plant growth, and maintain the balance of the ecosystem. This hidden infrastructure is as vital to the forest as the visible trees and understory plants, yet it operates entirely out of sight, a reminder that much of life's essential work happens in darkness and silence. In the Blue Mountains of Oregon lives something extraordinary. Hidden beneath the soil lies the largest known living organism on Earth, a single fungal network of honey mushroom that stretches for nearly 4 square miles. This colossal mycelium is thought to be thousands of years old, quietly growing and spreading while civilizations rose and fell above it. The organism was discovered in the late 1990s when scientists studying forest health in the region noticed patterns suggesting that many honey mushrooms growing across a vast area might be genetically identical. By collecting samples and analysing their DNA, researchers confirmed that what appeared to be many separate fungi were actually all part of one individual. The mycelium spreads through the soil, connecting mushrooms that fruit hundreds of meters apart, all of them growing from the same underground network. This massive fungus occupies roughly 2,400 acres, making it larger than 1,600 football fields. Its weight is estimated at hundreds or possibly thousands of tons, far exceeding that of the largest blue whale. While the mushrooms visible above ground are modest in size, the true scale of the organism lies in the vast web of filaments spreading through the forest floor, invisible but omnipresent. The honey mushroom grows by extending its mycelium through soil and into the roots of trees. It can act as both a decomposer, breaking down dead wood, and sometimes as a parasite, attacking living trees when they are weakened by stress or injury. Over thousands of years, this fungus has slowly expanded its territory, consuming organic matter and incorporating it into its ever-growing network. The process is gradual, measured in inches per year, but given enough time, those small increments add up to something immense. Determining the age of this organism is challenging because fungi do not have growth rings like trees. Scientists estimate its age by calculating how fast mycelium typically grows and working backward from the area it now covers. Based on these calculations, the Oregon honey mushroom may be between 2,000 and 8,000 years old, possibly older. It was already ancient when the Roman Empire rose and was still expanding when European explorers first reached North America. What makes this discovery particularly remarkable is that it challenges our intuitive understanding of what constitutes a single organism. We think of large creatures as whales or elephants, beings we can see and define clearly. But this fungus redefines scale. It is distributed across a landscape with no central body or brain, yet it is genetically unified, all parts descended from a single spore that germinated millennia ago. Every mushroom it produces is technically part of the same individual, like branches on an enormous tree. This is not an isolated case. Since the Oregon discovery, other massive fungal organisms have been found in different locations. In Michigan, another honey mushroom network covers over 37 acres and weighs an estimated 100 tons. In Switzerland, research has identified ancient mycelia that may be even older than their American counterparts. These findings suggest that truly enormous, long-lived fungi may be more common than previously thought, hidden beneath forests around the world. The implications are profound. These giant organisms play crucial roles in their ecosystems, recycling nutrients on a massive scale and influencing forest structure over centuries. They remind us that life operates at timescales and spatial scales that dwarf human experience. While we measure our lives in decades, this fungus has been growing steadily for thousands of years. While we see individual trees, it experiences the forest as a unified landscape, its mycelium touching thousands of roots and logs simultaneously. To stand in the Blue Mountains and know that beneath your feet lies a single living being of such antiquity and size is humbling. You cannot see it or hear it, but it is there, woven through every cubic meter of soil, connecting the forest in ways that remain largely mysterious even to scientists. It is a reminder that the earth holds wonders that exceed our ability to directly perceive them, vast and patient lives unfolding in the darkness beneath our feet. Though mushrooms have no brains or nervous systems, they send information through their thread-like networks, using electrical pulses. These signals resemble the firing of neurons in animals, carrying messages about resources, dangers, or environmental changes. Scientists studying these patterns have found rhythms that look almost like language, with frequency changes that may convey meaning. The discovery of electrical signaling in fungi is relatively recent, made possible by advances in sensitive measuring equipment. Researchers can now insert tiny electrodes into mycelium and detect voltage changes as they propagate through the fungal network. These pulses travel along the hyphae at varying speeds and frequencies, creating patterns that differ based on what the fungus encounters in its environment. When mycelium contacts a new food source, such as a piece of wood or a pocket of nutrients, the electrical activity in nearby hyphae changes. The pulses become more frequent, or their amplitude increases, suggesting that the fungus is responding to the discovery. Similar changes occur when the mycelium encounters obstacles, toxins, or damage to part of the network. These signals can travel considerable distances through the mycelium, potentially alerting distant parts of the organism to conditions encountered in one location. Some researchers have compared these electrical patterns to the action potentials that propagate along nerve cells in animals. While the mechanisms differ, both involve changes in ion concentrations across cell membranes, creating waves of electrical activity that can transmit information. The similarity raises intriguing questions about the nature of signaling and communication in organisms that lack central nervous systems. Analysis of fungal electrical activity has revealed patterns that appear surprisingly structured. Bursts of pulses cluster together in groups, with pauses between them, creating rhythms that some scientists suggest resemble words or syllables in language. While this interpretation remains speculative, it highlights the complexity of fungal signaling. The patterns are not random noise, but organized sequences that may encode different types of information. Through these signals, fungi may coordinate growth and resource allocation across their networks. A mycelium encountering abundant food in one area might send signals that cause other parts of the network to redirect growth in that direction. Conversely, signals from a damaged area might trigger defensive responses or redirect resources away from the affected region. This coordination allows the fungus to behave as a unified organism, despite lacking a brain or central control system. The implications extend to the mycorrhizal partnerships fungi form with plants. When plant roots connect to fungal networks, the electrical activity in the mycelium changes. Some researchers believe fungi may use these signals to communicate with their plant partners, though the details of such communication remain unclear. The possibility that plants and fungi exchange information through electrical signals adds another layer to the already complex relationships these organisms maintain. Studying fungal signaling also raises philosophical questions about consciousness and intelligence. If fungi can sense their environment, remember past experiences, and make decisions about resource allocation, do they possess a form of intelligence? The answer depends on how we define these terms. Fungi clearly lack consciousness in any way we would recognize, but they demonstrate sophisticated information processing that allows them to thrive in complex environments. They are problem solvers, optimizers, and in some sense decision makers, all without a single neuron. There is something profound about the idea of electrical signals coursing through soil, invisible conversations happening beneath every footstep. The forest floor is not silent but alive with transmissions. Fungi speaking in pulses and frequencies we are only beginning to understand. This hidden communication network operates constantly, coordinating the work of decomposition, partnership, and growth that sustains ecosystems above ground. When scientists tested what life could endure beyond Earth, they brought fungal spores along for the journey. To their amazement, some species not only survived exposure to the vacuum of space, but also endured intense radiation and freezing temperatures. These spores floated outside spacecraft for weeks, and when brought back to Earth, many were still capable of growing. These experiments were conducted during various space missions, with spores attached to the exterior of satellites and space stations. In the vacuum of space, there is no air, no pressure, and temperatures swing between extremes as the spacecraft moves in and out of sunlight. Cosmic radiation, far more intense than anything experienced on Earth's surface, bombards everything exposed to it. These conditions would kill most living things within minutes. Yet fungal spores proved remarkably resilient. Protected by their tough outer coats, many remained viable through weeks of exposure. When scientists retrieved them and placed them in suitable growing conditions, the spores germinated and produced healthy mycelium. The fungi had survived one of the harshest environments imaginable, demonstrating a toughness that challenges our assumptions about the limits of life. This resilience makes fungi fascinating candidates for astrobiology, the study of life beyond Earth. If fungal spores can survive in space, it raises the possibility that life could travel between planets or even between star systems. The theory of panspermia suggests that life might spread through the cosmos by hitching rides on meteorites, comets, or interstellar dust. Microorganisms, including fungal spores, could potentially survive such journeys, waiting dormant until they encounter conditions suitable for growth. The implications extend to human space exploration. As we venture beyond Earth, we will need to bring life support systems with us. Fungi could play crucial roles in these systems, breaking down waste, recycling nutrients, and possibly even producing food. Their ability to survive space conditions makes them valuable partners for long-duration missions, where resupply from Earth is impossible. Some researchers are exploring how fungi might help terraform other worlds. On Mars, for example, fungi could potentially break down rock into soil, just as they did on early Earth. they might help create habitable conditions for other organisms, starting with hardy lichens and eventually supporting more complex plant life. The same resilience that allows them to survive space could make them pioneers on new worlds, preparing the way for future ecosystems. The experiments also revealed interesting details about what protects fungal spores. Their walls contain melanin, the same pigment that colors human skin and hair. In fungi, melanin appears to provide protection against radiation, absorbing and dissipating the energy that would otherwise damage cellular structures. Spores with higher melanin content survived better than those with less, suggesting that pigmentation is a key factor in radiation resistance. Temperature extremes posed less of a challenge than expected. Many fungal species regularly experience freezing temperatures on Earth and have evolved mechanisms to protect their cells from ice damage. The cold of space, while more extreme, operates on similar principles. Fungi that can survive Arctic winters proved equally capable of enduring the cold of the void. Heat during sun exposure was more challenging, but the brief cycles between light and shadow prevented sustained heating that might cause damage. The vacuum itself, surprisingly, was not the greatest threat. While most living cells would rupture in vacuum as water boiled away, spores are designed to be dormant and desiccated. They contain very little water and are already adapted to survive long periods of dryness. The vacuum simply maintained them in their dormant state, neither causing additional damage nor awakening them. These findings suggest that fungal spores might already be traveling through space naturally. Every time a large meteor strikes Earth, it throws debris into space, some of which escapes Earth's gravity entirely. This debris could carry dormant spores, launching them on journeys across the solar system or beyond. If Earth has been seeding space with its microbes for billions of years, it is possible that life from our planet has already reached other worlds, waiting in dormant spores for the right conditions to awaken. There is something humbling about this possibility. The mushrooms we see in forests, so terrestrial and earthbound in appearance, might be part of a story that extends far beyond our planet. The spores they release into the air could, in the right circumstances, become travelers across cosmic distances, carrying the spark of life to places we have never imagined. In their quiet resilience, fungi remind us that life is tougher, more adaptable, and potentially more widespread than we once believed. In the aftermath of the nuclear event at Chernobyl in 1986, scientists studying the affected reactor made a remarkable discovery. Certain fungi were not just persisting in areas with elevated radiation, but appeared to grow toward the radiation itself. These remarkable organisms contain melanin, the same pigment found in human skin, which may allow them to convert radiation into chemical energy. The event at Chernobyl in 1986 created an area where radiation levels remain elevated even decades later. This has become an unexpected natural laboratory for studying how life adapts to challenging conditions. While the area requires caution for humans, various organisms have continued to exist there, and some have adapted in fascinating ways. Among the most surprising discoveries were fungi growing on surfaces inside the reactor building itself, species including certain molds and mushrooms colonized areas near the reactor core, places where radiation levels remained quite high. Rather than avoiding these locations, the fungi appeared drawn to them, growing more abundantly where radiation was present than in areas with lower levels. Researchers investigating this phenomenon found that many of these fungi contain high concentrations of melanin. This pigment, which provides color in everything from human skin to bird feathers, appears to have additional functions in these radioactive environments. Laboratory experiments showed that melanin-rich fungi could grow faster when exposed to radiation, suggesting they might be using the radiation as an energy source. The mechanism, called radiosynthesis, remains partially understood. It appears that melanin can capture energy from radiation and use it to drive chemical reactions, similar to how chlorophyll captures light energy during photosynthesis. The fungi convert the radiation's energy into chemical forms they can use for growth and metabolism. While the efficiency is lower than photosynthesis with sunlight, in environments with intense radiation and little organic matter, this ability provides a significant advantage. This discovery has profound implications. On Earth, nearly all life depends ultimately on photosynthesis, the conversion of sunlight into chemical energy by plants and algae. But in environments where sunlight cannot reach, such as deep caves, ocean floors, or the interior of structures with elevated radiation, alternative energy sources become available. Fungi that can use radiation open up entirely new possibilities for where and how life can exist. The potential applications are equally remarkable. Scientists are exploring whether these fungi could help remediate nuclear waste sites. By growing in contaminated soil or water, they could potentially reduce radiation levels while also stabilizing the environment for other organisms. Fungi might also protect astronauts during deep space missions, where cosmic radiation poses serious health risks. Cultivating radiation-eating fungi inside spacecraft or habitats could provide a biological shield, absorbing radiation that would otherwise harm humans. Research continues into the exact mechanisms and limits of this ability. Not all melanin-containing fungi show the same response to radiation, suggesting that additional factors are involved. Some species appear to have specific adaptations beyond melanin production that enhance their radiation tolerance. Understanding these adaptations could lead to new approaches for protecting both humans and ecosystems from radiation damage. Walking through the Chernobyl exclusion zone today, you might see mushrooms fruiting from soil, apparently unbothered by radiation levels that require caution for humans. Trees have grown back, birds nest in abandoned buildings, and fungi continue their patient work of decomposition. Life has found ways to persist and adapt, with fungi demonstrating remarkable resilience in challenging conditions. These environments with elevated radiation have become unexpected laboratories for studying life's resilience. The fungi in these areas demonstrate that organisms can adapt to conditions that challenge them. They show that energy comes in many forms, and that evolution finds solutions across a remarkable range of conditions. In places where radiation is present, mushrooms have found ways not just to persist, but to use it as a resource, demonstrating life's remarkable flexibility and adaptability. One of the most hopeful discoveries in recent years is that some fungi can break down materials that usually persist for centuries. Certain species have been found capable of digesting plastic and petroleum, transforming toxic waste into harmless substances. In experiments, fungi have turned oil-soaked soil into blooming fields within weeks. The problem of plastic pollution has grown to staggering proportions. Millions of tons accumulate in landfills and oceans, persisting for hundreds of years because few organisms can break down synthetic polymers. Petroleum spills contaminate soil and water, with cleanup efforts often proving expensive, slow, and incomplete. For decades, these pollutants seem destined to plague ecosystems indefinitely, resistant to natural decomposition. Then researchers began discovering fungi with unexpected abilities. In the Amazon rainforest, scientists found a species called Pestalothiopsis microspora that could digest polyurethane, one of the most common and persistent plastics. The fungus secreted enzymes capable of breaking the chemical bonds in plastic, converting the polymer into smaller molecules it could metabolize. Even more remarkably, it could do this in both oxygen-rich and oxygen-poor environments, making it potentially useful in landfills where oxygen levels are low. Other fungi showed similar abilities with different materials. Some could break down polystyrene, the foam plastic used in packaging. Others digested polyethylene, the plastic in bags and bottles. Each species produced specific enzymes tailored to attack particular polymer structures, suggesting that fungi have evolved diverse strategies for breaking down synthetic materials, or that they are adapting to utilize these novel carbon sources. Oil-degrading fungi have proven equally valuable. When petroleum contaminates soil, it forms toxic compounds that poison plants and animals while resisting natural breakdown. Certain mushrooms can colonize these contaminated areas, secreting enzymes that break down hydrocarbons into simpler, harmless molecules. The treated soil gradually returns to health, becoming capable of supporting plant growth again. Field trials have demonstrated this potential. In oil-contaminated areas, researchers have introduced fungal mycelium either as spawn mixed into soil or as mats laid over affected surfaces. Within weeks to months, oil concentrations dropped dramatically. Plants began growing where they previously could not survive. Birds and insects returned as the ecosystem recovered. The fungi essentially ate the pollution, transforming it from poison into nutrients. This approach, called mycoremediation, offers several advantages over traditional cleanup methods. It is relatively inexpensive, requiring mainly fungal spawn and time rather than heavy equipment and harsh chemicals. It works in place, without needing to excavate and transport contaminated soil. And it is sustainable, using natural biological processes rather than creating additional environmental impacts. The fungi do the work, quietly and efficiently, breaking down pollutants while supporting ecosystem recovery. Researchers are now exploring how to optimize these processes. Different fungal species work best on different pollutants, so selecting the right species for each situation is crucial. Environmental conditions like temperature, moisture, and pH affect how quickly fungi can break down contaminants. Understanding these factors allows scientists to create conditions that maximize the fungi's effectiveness. the potential applications extend beyond soil remediation. Scientists are investigating whether fungi could help clean contaminated water, break down plastic in oceans, or process waste at landfills. Some envision large-scale facilities where fungi actively digest plastic waste, turning problems into resources. The enzymes themselves might be harvested and used in industrial processes, accelerating plastic recycling or breaking down materials that currently cannot be recycled. You don't need to hold on to these details. Let them drift past you like spores on the wind. The important thing is the sense of possibility, the way nature offers solutions to problems we created. Fungi have been breaking down organic matter for hundreds of millions of years, and they are now turning those ancient abilities toward modern challenges, teaching us that healing often comes not from human invention alone, but from partnership with the organisms that have always worked to keep Earth healthy. Among the many varieties of mushrooms, some require careful identification and are best left alone, while others offer flavors so striking they mimic lobster or chicken almost perfectly, becoming treasured foods across cultures. The boundary between what's safe to eat and what should be avoided makes knowledge and caution essential for foragers. The boundary between edible and inedible in the fungal kingdom can be subtle and often invisible to casual observers. Some mushrooms that should be avoided look remarkably similar to edible species, which is why expert guidance is essential for foragers. The Amanita family, for example, includes both species to avoid entirely and others that experienced foragers work with safely. This is why learning from experts and never consuming a mushroom without absolute certainty is the foundation of safe foraging. This is why foraging communities emphasize education, patience, and learning from experienced guides. Many regions offer foraging courses where you can learn to identify species safely, understanding both what to seek and what to avoid. The respect foragers show toward fungi reflects generations of accumulated wisdom about these remarkable organisms. Throughout history, certain mushroom species have taught humanity important lessons about the need for careful identification. This knowledge, passed through generations, has created the strong tradition of caution and expertise that defines responsible foraging today. Fungi have evolved remarkable chemical diversity as natural defenses, which is part of what makes them such fascinating organisms to study. Let's turn our attention now to the delicious varieties that have become culinary treasures. Yet the fungal kingdom also offers some of the most prized flavors in cuisine. The chicken of the woods mushroom Latiparus grows on tree trunks in shelf-like formations of bright yellow and orange. When cooked, its texture and flavor resemble chicken breast to a remarkable degree. It tears apart in similar chunks and absorbs seasonings in much the same way. Vegetarians and vegans prize it as a meat substitute that occurs naturally in the wild. The lobster mushroom, Hypomyces lactiflorum, is actually a parasitic fungus that transforms other mushrooms, colouring them bright red-orange and giving them a firm texture and subtle seafood flavour. Despite its name, it is completely unrelated to lobsters. Yet the taste resemblance is striking enough that chefs incorporate it into dishes as they would actual shellfish. This convergent flavour evolution, where unrelated organisms develop similar tastes, demonstrates the surprising diversity of chemical compounds fungi can produce. Lion's mane mushrooms offer yet another texture and flavour, with a delicate, slightly sweet taste and a texture some compare to crab or fish when properly prepared. Morels, highly prized and expensive, have a rich, earthy, nutty flavour that chefs celebrate. Chanterelles bring fruity, peppery notes. Each edible species contributes its unique combination of flavour compounds, expanding the palette available to cooks. This contrast in the fungal kingdom underscores an important truth about fungi. They are master chemists, producing thousands of different compounds, some beneficial, some neutral, some requiring caution. Evolution has shaped this chemical diversity over millions of years. What we perceive as delicious flavour or compounds to avoid are simply different points on a spectrum of chemical possibility. For foragers, this means caution is essential. Every mushroom should be identified with certainty before consumption. Field guides, expert knowledge, and careful observation all play roles in safe foraging. The risks are real, but so are the rewards for those who learn to navigate the fungal kingdom with respect and care. In this balance between danger and delight, mushrooms remind us that nature's offerings come with responsibilities, and that wisdom lies in knowing the difference between what nourishes and what harms. For thousands of years, mushrooms have held a place not only in kitchens, but also in temples and ceremonies. Ancient peoples used certain fungi in spiritual rituals, believing they opened doorways to altered states and divine wisdom. Cave paintings thousands of years old show figures holding mushrooms, suggesting this relationship stretches back to the dawn of human culture. In Siberia, shamans used the red and white Amanita muscaria, the iconic mushroom of fairy tales, in ceremonies designed to induce trance states and spiritual visions. They prepared the mushroom carefully, often drying it to reduce certain toxic compounds while preserving others that produced the desired effects. During winter solstice celebrations and healing rituals, shamans would consume the mushroom and enter altered states, returning with insights they believed came from the spirit world. In Mesoamerica, the Aztecs referred to sacred mushrooms as Teonanacatl, meaning flesh of the gods. Psilocybin-containing mushrooms grew naturally in the mountains and forests of central Mexico, and indigenous peoples incorporated them into religious ceremonies long before European contact. Spanish conquistadors documented these practices with mixture of fascination and horror, recognizing that the mushrooms played central roles in indigenous spirituality. The ceremonies typically occurred at night, guided by experienced practitioners who understood dosage, preparation, and ritual context. Participants sought healing, divination, or connection with ancestors and deities. The mushrooms were not used casually but treated with profound respect, as sacred tools that demanded proper reverence. This careful ceremonial use contrasts with modern recreational approaches, reflecting different cultural understandings of these fungi's power and purpose. Archaeological evidence suggests mushroom use in spiritual context extends even further back. Rock paintings in Spain and Algeria, dating to perhaps 7,000 years ago, depict human figures holding mushroom-shaped objects. Some researchers interpret these as evidence of prehistoric mushroom ceremonies, though the interpretation remains debated. Regardless, the antiquity of these images suggests that humans have recognized something special in certain mushrooms for most of our history as a species. The chemical compounds responsible for these effects are now well understood. Psilocybin and psilocin, found in many sacred mushrooms, interact with serotonin receptors in the brain, producing profound changes in perception, emotion, and thought. Research has shown these compounds can temporarily increase connectivity between brain regions that normally communicate less frequently, possibly explaining the sense of insight and connection many users report. In recent decades, scientific interest in these compounds has revived. Medical researchers are exploring their potential to treat depression, anxiety, addiction, and post-traumatic stress. Clinical trials have shown promising results, with some patients experiencing lasting improvements after carefully guided sessions with psilocybin. This modern research echoes ancient understanding, suggesting that these mushrooms may indeed have therapeutic value when used appropriately and with proper support. Indigenous traditions have continued despite centuries of suppression. In Mexico, some communities have maintained ceremonial mushroom use through generations, preserving knowledge and practices that might otherwise have been lost. These traditions offer insights into sustainable, respectful relationships with powerful natural substances, emphasizing community, guidance, and spiritual context rather than individual pursuit of novel experiences. The mushroom's role in human spirituality reflects something deeper about how we relate to the natural world. Throughout history, people have sought meaning, healing, and transcendence through connection with other organisms. Plants, fungi, and even animals have served as bridges between ordinary consciousness and experiences perceived as sacred or revelatory. Mushrooms, with their sudden appearance and their chemical effects, seemed particularly suited to this role. To understand these historical uses is not to recommend them. The mushrooms carry risks, both medical and psychological, especially when used without proper knowledge, preparation, or support. But their place in human history reminds us that the fungal kingdom has shaped not only ecology, but also culture, influencing art, religion, and medicine across millennia. The mushrooms growing quietly in forests have been witnesses and participants in humanity's long search for meaning, offering their mysterious compounds to those who approached them with reverence and care. One of the most remarkable phenomena in nature involves fungi that form intricate relationships with insects. Cordyceps fungi develop connections with ants and other insects, eventually influencing their movements in fascinating ways. The affected insect climbs to a specific height and position, where the fungus can release spores from an ideal location for dispersal. The process begins when a spore lands on an insect's body. The fungus develops thread-like hyphae that extend into the insect's interior. These threads spread through the body while the insect remains mobile, which allows the fungus's strategy to work effectively. The relationship is precisely timed, with the fungus coordinating its growth with the insect's movements. As the relationship progresses, something remarkable happens. The fungus begins to guide the insect's behavior in specific ways. The ant moves away from its colony and familiar territory, eventually climbing upward onto vegetation at a particular height above the ground. This height is not random, but carefully calibrated for optimal spore dispersal, high enough that air currents can carry spores over a distance, but not so high that they scatter too widely to find new hosts. When the ant reaches the appropriate location, it secures itself to a leaf or stem with its mandibles. This attachment is remarkably strong and stable. The fungus has guided its host to precisely the right place. Over the following hours or days, the fungus continues its development. A stalk grows from the ant's body, usually emerging from the head or thorax, and develops a spore-bearing structure at its tip. The stalk positions the spores above the ant's corpse, in open air where wind can catch them. When mature, the spores are released in enormous numbers, drifting down to the forest floor below where other ants travel. The cycle begins again as spores land on new hosts. The entire strategy is precisely calibrated, from the timing of infection to the height of the climb to the angle of the stalk, all evolved to maximize the fungus's reproductive success. Different cordyceps species have evolved to parasitize different insects. Some specialize in ants, others in beetles, caterpillars, or even spiders. Each has its own particular manipulation strategy, guiding its host to locations and positions that suit its spore dispersal needs. Some hosts climb high into trees, others attach to the undersides of leaves. The specificity is remarkable, suggesting millions of years of coevolution between parasites and hosts. Researchers studying these fungi have discovered that the mechanism of behavioral influence remains partially mysterious. The fungus doesn't affect the brain directly, but instead appears to work through chemical signals that interact with muscles and neural pathways. It may produce compounds similar to the insect's own neurotransmitters, working within the systems that control movement and behavior. The precision of this influence guiding complex behaviors like climbing and securing to vegetation suggests sophisticated biochemical communication. This phenomenon has captured scientific and popular imagination alike, demonstrating nature's remarkable strategies for reproduction and dispersal. For the ants and other insects involved, this represents an ongoing evolutionary relationship. Some ant colonies have developed responses, with workers carrying affected individuals away from the nest. This long relationship between fungus and insect has continued for millions of years, shaping both participants through natural selection. For humans, cordyceps offers more than just fascinating biology. Some species have been used in traditional medicine for centuries, particularly in China where Cordyceps sinensis grows in the Himalayan region. While these medicinal species do not infect humans, they contain compounds that may have therapeutic effects. Modern research is exploring potential applications, from immune support to athletic performance enhancement. The process of an ant being guided to climb and position itself as a platform for spore dispersal represents one of nature's many remarkable strategies. Like so many relationships in nature, it demonstrates the intricate ways organisms have evolved to interact with one another. Every breath you take carries with it tiny fragments of the fungal kingdom. Mushroom spores, the microscopic seeds of fungi, are so abundant that billions swirl invisibly through the air around us at any moment. They drift through cities and forests, floating on wind currents that can carry them for hundreds of miles. Scientists estimate that each cubic meter of outdoor air contains thousands of spores at any given time. In forests during active fruiting seasons, the concentrations can be even higher, with some measurements recording hundreds of thousands of spores per cubic meter. These spores are so small, typically between 3 and 20 micrometers in diameter, that they remain suspended in air for extended periods, settling only slowly under gravity. You cannot see them, but they are there with every breath. Your lungs have evolved to handle this constant influx. Mucus lining your airways traps most spores before they reach the deepest parts of your lungs, and your immune system recognizes and neutralizes any that do penetrate further. For most people, this continuous exposure to fungal spores causes no harm. We have breathed them since birth, and our bodies are adapted to their presence. The spores come from everywhere. Every mushroom releasing spores adds to the aerial population. Fungi fruiting on trees, in soil, on dead leaves, even growing on buildings all contribute. Some species release spores continuously, while others produce them in pulses tied to humidity, temperature, or time of day. The result is a constantly shifting population of airborne spores, mixing and moving with weather patterns. Rain affects spore concentrations in complex ways. Heavy rain can wash spores out of the air, temporarily reducing their numbers. But rain also triggers fruiting in many fungal species, so a few hours after a storm ends, spore concentrations often surge as freshly formed mushrooms release their reproductive cells. This pulse of spores following rain is part of why the air smells different after a storm. A scent that includes compounds released by fungi along with their spores. Wind carries spores across remarkable distances. Spores released in one location can travel hundreds or thousands of kilometers before settling. This explains how fungi can appear in isolated habitats soon after those habitats become available. When volcanic islands emerge from the ocean, fungi are among the first colonizers, their spores having drifted on winds from distant lands. When forests are cleared and then abandoned, fungal species from surrounding areas quickly establish themselves, their spores having been present in the air all along, waiting for suitable conditions. The global movement of spores connects ecosystems worldwide. Fungi from one continent can, given the right wind patterns and enough time, reach another continent entirely. This creates a kind of aerial web, linking distant forests, fields, and gardens through the invisible flow of spores. The same air that carries pollen and dust also carries the next generation of fungi, spreading them across the planet. Seasonal patterns affect which spores are most abundant. In temperate regions, autumn brings a surge of mushroom fruiting and corresponding increases in airborne spores. Spring may see different species contributing to the aerial population. These seasonal shifts create rhythms in the invisible spore population that mirror the visible changes in fungal fruiting bodies. Indoor air contains spores too, though usually in lower concentrations than outdoors. Spores enter through open windows and doors, on clothing, and attach to pets. Some fungi even grow indoors. particularly in damp areas, adding their spores to the indoor environment. For most people, this is harmless background noise. Only those with specific allergies or immune conditions need to monitor indoor spore levels carefully. To know that every breath connects you to this hidden kingdom can change how you think about air itself. What seems empty is actually filled with life in its smallest, most patient form. Each spore is a potential new fungus, waiting to land somewhere suitable, to germinate, to grow. Most will never find the right conditions, but enough do that the cycle continues, generation after generation, filling the air with invisible possibility. Unlike seeds, which are often heavy and bound to soil, fungal spores are designed for long-distance travel. Many are so tiny and lightweight they can remain airborne for remarkable lengths of time. Some have been detected more than 20 kilometers above Earth's surface, floating in the stratosphere where they may ride high-altitude winds across continents. The stratosphere is the layer of atmosphere extending from about 10 to 50 kilometers above Earth's surface. It is a realm of thin air and extreme conditions, far above where weather occurs, where commercial aircraft fly, and where most life exists. Temperatures can reach -60 degrees Celsius. Oxygen levels are too low to support animal life. Ultraviolet radiation from the sun, unfiltered by the thicker atmosphere below, is intense enough to damage most biological molecules. Yet fungal spores have been found here, collected by aircraft and balloons designed to sample the upper atmosphere. The spores are intact, protected by their tough outer walls from the cold, radiation, and desiccation. How they reach such heights is partly explained by their extreme lightness and small size. The tiniest spores can be caught in updrafts from storms or thermal currents that carry them upward, far beyond the altitude where they were released. Once in the stratosphere, spores can remain aloft for extended periods. With little gravity to pull them down in the thin air, and no rain to wash them out, they drift with high-altitude winds that circle the globe. These winds can reach speeds of hundreds of kilometers per hour, moving air and everything suspended in it across vast distances. A spore lifted into the stratosphere over one continent might circle the Earth before eventually descending over another continent entirely. This high-altitude transport has important implications for understanding global fungal distribution. Species once thought to have limited ranges may actually disperse their spores worldwide through stratospheric travel. This could explain how the same fungal species appear on multiple continents, even in locations separated by thousands of kilometers of ocean. The atmosphere itself serves as a highway for fungal dispersal at a truly planetary scale. The ability to survive in the stratosphere demonstrates remarkable resilience. The spores experience temperature extremes, intense UV radiation, and complete dryness. Their protective coatings shield the genetic material and cellular machinery inside, maintaining viability even after weeks or months of exposure. When these spores eventually descend to Earth's surface, many remain capable of germinating if they land in suitable conditions. This stratospheric population of spores may also play a role in atmospheric processes. Spores can serve as nuclei around which water droplets or ice crystals form, potentially influencing cloud formation and precipitation patterns. While research in this area is ongoing, it suggests that fungi may affect not just terrestrial ecosystems, but atmospheric processes at a global scale. The invisible presence of spores in the upper atmosphere could be participating in the water cycle itself. Scientists continue to investigate how common stratospheric spores are and which species reach these heights most frequently. Sampling is challenging, requiring specialized equipment and careful handling to avoid contamination. But the studies completed so far consistently find viable spores at remarkable altitudes, suggesting this is a normal, ongoing phenomenon rather than a rare occurrence. The journey from mushroom to stratosphere and back to Earth represents an extreme form of dispersal. Most spores fall near where they were released, settling within meters or kilometers of their parent fungus. But the tiny fraction that reaches the upper atmosphere becomes part of a global distribution network, potentially travelling farther in a few days than terrestrial organisms could move in thousands of years. There is something both humbling and uplifting about this knowledge. The mushrooms fruiting after rain are not just local events, but participants in a planetary system of dispersal. Their spores join an aerial community that circles the Earth, connecting ecosystems and spreading genetic diversity across continents. In their microscopic journeys, spores demonstrate that even the smallest life forms can traverse the globe, carried by winds we cannot see, through altitudes we rarely consider, linking forests and fields in ways that remain largely invisible to us. We often think of stone as unchanging, but even rocks yield to the patient persistence of fungi. Some species secrete acids that can slowly dissolve minerals, gradually transforming solid rock into fertile soil over hundreds or thousands of years. This quiet work prepared the way for plants to colonize land hundreds of millions of years ago. The process begins when fungal spores land on bare rock and germinate. The developing hyphae extend across the stone surface and into tiny cracks and pores. As they grow, they release organic acids, weak but persistent chemicals that react with minerals in the rock. These acids slowly dissolve the mineral structures, breaking apart the chemical bonds that hold stone together. Different minerals respond differently to fungal acids. Some, like limestone, dissolve relatively easily. Others, like granite, resist more stubbornly but still yield given enough time. The acids work their way along crystal boundaries and into microscopic fractures, gradually loosening the rock's structure grain by grain. What was solid becomes powder, and what was sterile stone becomes material that can support life. Lichens partnerships between fungi and algae are particularly effective at this process. The fungal partner provides structure and secretes acids, while the algal partner produces sugars through photosynthesis, feeding the fungus and allowing it to maintain its work even on bare rock. Together they create small patches of life on seemingly lifeless surfaces. Over decades and centuries, these patches expand, etching deeper into the stone. As the rock breaks down, it mixes with dead fungal tissue, trapped dust, and other organic matter to form primitive soil. This early soil is thin and poor compared to established forest soil, but it is enough for pioneer plants like mosses to take root, These plants accelerate the process, their roots working with fungi to further break down rock while their growth adds more organic matter to the developing soil. The transformation from rock to soil is measured in centuries or millennia, but it is relentless. Ancient rock surfaces gradually become coated with soil where forests eventually grow. Mountain ranges slowly weather away, their stone converted into the rich earth of valleys. The process continues everywhere. Rock is exposed to air, water, and life, with fungi playing a central role throughout. Evidence of this work is visible on old stonework. Tombstones in centuries-old cemeteries show patterns of erosion, where lichens and fungi have colonized their surfaces. Ancient stone monuments develop characteristic weathering patterns influenced by biological activity. Even modern concrete structures show signs of fungal colonization, with acids gradually affecting the surface chemistry of the material. Scientists studying early Earth history believe fungi were crucial to making the planet habitable for complex life. When plants first began colonizing land around 450 million years ago, they faced bare rock surfaces with virtually no soil. Fungi had already been working on these surfaces for hundreds of millions of years, slowly creating the first thin layers of weathered material that plants could use. This partnership between fungi and early plants accelerated soil formation, transforming rocky landscapes into the green world we know today. The same process continues in modern landscapes wherever rock is exposed. After glaciers retreat, leaving bare rock behind, fungi and lichens are among the first colonizers. After volcanic eruptions create new rock surfaces, these organisms begin their patient work within years or decades. They are the advance guard of life, preparing the ground for everything that follows. In gardens and fields, this process works alongside human soil management. Mycorrhizal fungi, which partner with plant roots, continue to break down mineral particles in soil, releasing nutrients plants need. The relationship between fungi and soil formation is not just ancient history, but an ongoing process that maintains soil fertility. Without fungi constantly working to break down rock particles and recycle organic matter, soils would gradually lose their fertility and structure. To stand on fertile ground is to stand on stone that fungi help transform. The rich earth supporting forests and farms is partly the product of fungal acids, dissolving rock over vast spans of time. Every handful of soil contains rock fragments in various stages of weathering, with fungi still working on the process. In their patient, invisible way, fungi are constantly remaking the Earth's surface, turning the bones of the planet into the skin that supports all terrestrial life. Step into a grassy field after rain, and you may find a perfect circle of mushrooms as though left behind by dancing fairies. These fairy rings form as a single fungus spreads outward in all directions from a central point. The mushrooms you see mark the advancing edge of the hidden mycelium, with some rings growing larger each year for decades or even centuries. The process begins with a single spore germinating in one spot. The mycelium grows outward from this central point, extending its threads in all directions at roughly the same rate. Because the fungus is expanding equally in all directions from a central origin, the result is a circle. As the mycelium reaches the edge of its expanding territory, it produces mushrooms, creating a visible ring that marks the frontier of its underground empire. Year after year, the mycelium continues spreading outward. The center of the ring, where the fungus first grew, may become depleted of nutrients, so the mycelium there dies back or becomes less active. The living edge keeps advancing, and each fruiting season produces mushrooms in a larger ring. By measuring the ring diameter and knowing the species' typical growth rate, scientists can estimate how old the fairy ring is. Some fairy rings are centuries old. The mycelium has been expanding continuously through multiple human lifetimes, slowly claiming more territory. The largest known rings span more than 0.5 kilometer in diameter, representing fungi that may have been growing for 600 years or more. These ancient rings were already expanding when medieval farmers first ploughed their fields, and they continue their patient growth today. The grass inside and around fairy rings often shows distinct patterns. Sometimes the grass grows more vigorously where mushrooms fruit, greener and taller than surrounding areas, nourished by nutrients the fungus makes available. Other times, the grass may die back in the active zone where mycelium is most dense, creating a bare or brown ring. These patterns change through the seasons and years, creating dynamic landscapes within the larger pattern. Folklore around the world has interpreted fairy rings as magical or supernatural. European traditions held that fairies danced in circles at night, and mushrooms grew where their feet touched the ground. Stepping into a fairy ring was thought to trap you in the fairy realm or bring bad luck. In other traditions, fairy rings were portals to other worlds, or signs of buried treasure. The perfect geometry of the rings seemed too precise to be natural, demanding supernatural explanation. Even knowing the biological mechanism, there is something enchanting about discovering a fairy ring. The geometry is so exact, the spacing of mushrooms so regular, that the effect feels intentional. To stand in the center of a large ring and know that you are surrounded by a single organism spreading steadily outward from beneath your feet creates a sense of connection to something vast and patient. Not all fairy rings are perfectly circular. Obstacles like trees, rocks, or areas of unsuitable soil can distort the shape, creating arcs or incomplete circles. Multiple fairy rings may intersect, creating complex patterns as different mycelia encounter each other. Some species produce arcs or scattered patterns rather than rings, depending on their growth habits and the environment they inhabit. Fairy rings also provide valuable information to mycologists by studying ring size and growth rate. Researchers can learn about fungal longevity and dispersal patterns. Some species known only from fairy rings may produce mushrooms so rarely that the rings themselves are more reliable indicators of the fungus's presence than the mushrooms. The rings become living monuments to persistence, marking where fungi have claimed territory over human-scale time periods. Allow yourself to simply let these thoughts drift. You don't need to hold on to the details, just the feeling of these patterns and cycles. Even now, somewhere, fairy rings are expanding by millimeters, mushrooms are releasing spores into the night air, and the quiet work of fungi continues in darkness and stillness, patient as stone, gentle as rain. The fungal kingdom spans an extraordinary range of sizes. Some mushrooms are so tiny they fit on a pinhead, visible only under magnification. Others, like giant puffballs, can grow as large as beach balls, weighing more than 20 pounds when mature. This range from nearly invisible to monumentally large reminds us that fungi exist at every scale. At the smallest end of this spectrum are mushrooms so minute they escape notice entirely. These microfungi sprout from decaying leaves and twigs, their caps measuring less than a millimetre across. Under a magnifying glass or microscope, they reveal delicate structures. Tiny gills or pores arrange with the same precision as their larger relatives. Each is a complete mushroom in miniature, releasing spores and performing the same ecological work as species hundreds of times their size. These microscopic mushrooms often grow in clusters, dozens or hundreds together, creating what might look like a dusting of powder on a dead leaf. Only close inspection reveals that each speck is an individual fungus, complete with stalk, cap, and spore-bearing surface. They fruit, release spores, and decompose in a matter of days, their entire visible existence compressed into a brief window barely noticeable to humans passing by. In the middle ranges are the mushrooms most familiar to us: button mushrooms in grocery stores, chanterelles in forests, toadstools in fairy tales. These measure from a few centimetres to perhaps 20 or 30 centimetres across. They are substantial enough to notice, to photograph, to gather, but still small enough to hold comfortably in a hand. This is the scale at which humans most commonly interact with fungi, the size that matches our perception and attention. Then there are the giants. Giant puffballs, Calvatia gigantea, can reach diameters of over 50 centimetres and weights exceeding 20 kilograms. When fully mature, a single specimen might weigh as much as a small child. These white spheres emerge from soil seemingly overnight, swelling to impressive proportions within days. Their interior tissue, when young, is pure white and firm, gradually transforming into a mass of olive-brown spores as the mushroom matures. The sheer volume of spores a giant puffball produces is staggering. A large specimen might contain 7 trillion spores or more, each one a potential new fungus. When the puffball's skin breaks open or is disturbed, these spores release in vast clouds that billow into the air like smoke. The evolutionary strategy is clear: produce such enormous numbers that even if only a tiny fraction of spores find suitable conditions, the species survives and spreads. Other large mushrooms grow not as spheres but as shelves or brackets on tree trunks. Some bracket fungi develop fruiting bodies that can span more than a metre across and weigh several kilograms. These persistent structures may last for years, adding new layers of spore-bearing tissue each season. They are monuments to patience, growing slowly but steadily on the sides of living or dead trees, visible from a distance as substantial growths that alter the tree's silhouette. The range in size reflects the diversity of fungal strategies. Tiny mushrooms exploit small, ephemeral resources, fruiting quickly before their substrate dries or decays completely. Giant mushrooms draw on extensive underground networks and abundant resources, producing massive sporulating structures that maximize dispersal range and spore numbers. Between these extremes lie countless variations, each adapted to particular ecological niches and resource availability. This size diversity means that a forest contains mushrooms at every scale simultaneously. Walk through woodland and you pass giants without seeing the miniatures at your feet. Bend close to examine a rotting leaf, and you discover whole communities of microscopic fungi you never knew existed. Use a hand lens or microscope, and even tinier species reveal themselves, occupying ecological spaces measured in millimeters. The forest is layered not just in space but in scale, with fungal life operating at dimensions from the microscopic to the monumental. For humans, this range challenges our ability to perceive the full fungal kingdom. We notice what matches our scale, but we miss most of what exists above and below that narrow band. The truly small require tools to see. The truly large may be too big to recognize as single organisms, their extent hidden underground or spread across landscapes. Only by expanding our perspective to encompass the full range can we begin to appreciate how thoroughly fungi permeate every terrestrial ecosystem. The contrast between smallest and largest also reminds us that size is not a measure of importance. The tiniest mushrooms play roles just as crucial as the giants, each decomposing their particular substrates, each contributing to nutrient cycles. Size is simply another dimension of diversity, another axis along which evolution has explored possibilities. In the fungal kingdom, success comes in every size, from dust speck to boulder, each perfectly suited to its particular way of living. Truffles, prized underground mushrooms, emit an aroma so powerful it captivates both humans and animals. Their scent contains compounds similar to natural signals many creatures use for communication. For pigs especially, the smell of truffles is nearly irresistible, which is why farmers once relied on them to locate these hidden treasures. Truffles grow entirely underground, forming their fruiting bodies in soil near tree roots with which they maintain mycorrhizal partnerships. Unlike mushrooms that push above ground to release spores into air, truffles rely on animals to find them, dig them up, and eat them. The spores pass through the animal's digestive system unharmed and are deposited elsewhere in droppings, completing the dispersal cycle. For this strategy to work, truffles must be detectable underground, which is where their powerful aroma becomes essential. The scent of truffles is complex, composed of dozens of volatile compounds that waft through soil pores and rise to the surface. Some of these compounds resemble pheromones, chemical signals animals use for reproduction and communication. This molecular mimicry may have evolved specifically to attract mammals, particularly those that dig and forage in soil. The aroma triggers responses in animal brains, creating attraction that compels them to search for the source. Pigs are famously sensitive to truffle scent. Female pigs especially respond strongly because some truffle compounds resemble pheromones produced by male pigs. A pig can detect truffles buried 30 centimeters or more below the surface, precisely locating them through scent alone. Historically, truffle hunters trained pigs to find truffles, following them through forests and digging where the pig indicated. The challenge was preventing the pig from eating the truffle itself once found, as their enthusiasm for the scent translated into eagerness to consume the source. Dogs have largely replaced pigs for truffle hunting in modern times. They can be trained to find truffles without being as motivated to eat them, making them more manageable partners. Different dog breeds work, but all require training to recognize the scent and indicate finds without destroying them. The partnership between humans and truffle hunting dogs is centuries old in some European regions, with knowledge passed through generations of hunters. For humans, truffle aroma is equally compelling, though for different reasons. The scent is earthy, musky, and intensely savory, with notes that people describe as garlicky, mushroomy, or reminiscent of aged cheese. This complex aroma enhances dishes even in tiny amounts, with just a few grams of shaved truffle transforming the flavor profile of pasta, eggs, or risotto. The compounds responsible are volatile, dissipating quickly once the truffle is cut, which is why truffles are typically used fresh and stored carefully to preserve their aroma. Different truffle species produce different aromatic profiles. Black truffles, primarily Tuber melanosporum, offer chocolate and earthy notes. White truffles, Tuber magnatum, provide more garlicky, pungent aromatics. Summer truffles are milder, while winter varieties can be more intense. Each has its devotees, and prices reflect both scarcity and demand, with the finest specimens commanding hundreds of dollars per ounce. The difficulty of cultivating truffles adds to their mystique and value. While some success has been achieved in establishing truffle orchards where trees are inoculated with truffle mycelium, the process remains unpredictable. Truffles require specific soil conditions, climate, and tree partners to form. Even in ideal conditions, it may take 7 to 10 years before trees begin producing truffles, and yields vary dramatically from year to year. This unpredictability means wild truffles, found through traditional hunting methods, remain important and valuable. The economics of truffle hunting have created entire regional industries. In parts of France, Italy, and Spain, truffle season brings hunters and their dogs into forests under strict traditional rules. The locations of productive truffle grounds are closely guarded secrets, sometimes passed through families for generations. Markets devoted entirely to truffle sales operate during peak season, with buyers examining and smelling specimens to assess quality before negotiating prices. Beyond their culinary value, truffles represent a remarkable evolutionary adaptation. By growing underground and producing irresistible scents, they have turned mammals into dispersal agents. The strategy works so well that truffles have evolved multiple times in different fungal lineages, suggesting that going underground and becoming aromatic is a successful approach when conditions favour it. Each truffle species has refined its particular scent profile, optimizing the blend of compounds that attracts its preferred animal partners. To walk through a forest knowing that beneath your feet truffles may be developing, releasing their complex aromatics into soil, is to appreciate another hidden dimension of fungal life. These organisms never see daylight yet have evolved strategies as sophisticated as any above-ground mushroom. Their invisible presence, detectable only through scent, reminds us that the soil is not inert but alive with fungi pursuing their own strategies for survival and reproduction. Deep in forests, trees are connected through fungal networks that allow them to share resources and information. Through these underground partnerships, a shaded tree may receive sugars from sunlit neighbours, passed along by fungal threads. If threats arise, chemical signals can spread through the network, alerting other trees to prepare their defences. This phenomenon, sometimes called the Wood Wide Web, represents one of nature's most intricate partnership systems. The fungal networks that create these connections are mycorrhizal associations, partnerships where fungal hyphae interface directly with tree roots. The fungi receive sugars and other carbon compounds from the trees, produced through photosynthesis. In return, the fungi provide the trees with water and nutrients, particularly phosphorus and nitrogen, gathered from soil far beyond where roots can reach. But the relationship goes deeper than simple exchange. The fungal network connects multiple trees, sometimes dozens or hundreds of individuals. A large tree with abundant access to sunlight produces more sugars than it needs for itself. Rather than simply storing the excess, it can transfer some through the fungal network to neighbouring trees that are struggling. Seedlings growing in deep shade, unable to photosynthesize efficiently, receive life-sustaining sugars through these underground connections, allowing them to survive until gaps in the canopy give them access to more light. This resource sharing is not random charity but a system that benefits the forest as a whole. By keeping more trees alive, the network maintains forest health and resilience. Young trees that survive through shared resources become the next generation of canopy trees when older individuals die. The system creates continuity, ensuring that the forest persists even as individual trees come and go. The networks also transmit information. When a tree is attacked by insects, it can produce defensive compounds and release chemical signals through its roots into the fungal network. These signals travel through the mycelium to neighbouring trees, which detect them and activate their own defences in preparation for possible attack. This early warning system allows trees to protect themselves before insects arrive, giving them a defensive advantage. Research has shown that trees can even recognize their kin through these networks. Some species allocate more resources to genetically related neighbors than to strangers, suggesting that the networks carry information about identity as well as need. How trees sense this relatedness through fungal intermediaries remains a subject of active research. But the phenomenon demonstrates that these connections facilitate complex interactions we are only beginning to understand. The fungal networks themselves are not passive conduits, but active participants in forest dynamics. The fungi can direct resources preferentially towards certain trees or retain nutrients for their own growth when conditions favour it. They are partners with their own interests, not merely servants of the trees. The relationship is mutualistic, benefiting both parties, but also competitive in subtle ways. with each partner maximizing its own success while maintaining the cooperation that makes survival possible. Different tree species may connect to the same fungal networks, creating interspecies communities underground. A Douglas fir might share network connections with a paper birch, allowing resource transfer between completely different types of trees. In some forests, the networks are so extensive that nearly every tree is connected, directly or indirectly, to every other tree, creating a forest-scale superorganism where information and resources flow according to complex rules we are only beginning to decode. Old trees, sometimes called mother trees, appear to play central roles in these networks. They have the most extensive root systems and the most established fungal connections, making them hubs in the underground web. When these old trees die, the network reorganizes, with other large trees gradually assuming hub roles. The death of a major hub can affect forest health across a wide area, as the network loses a key node in its resource distribution system. Understanding the wood wide web has changed how foresters and ecologists think about forest management. Clear cutting, which removes all trees from an area, destroys not just the visible trees but the underground networks that supported them. Replanting in cleared areas means starting from zero, with young trees lacking the network connections that would help them establish. More selective forestry that maintains some mature trees preserves the networks, allowing regeneration to proceed more naturally. The existence of these networks also raises philosophical questions about individuality and community in nature. Is a forest a collection of individual trees competing for resources, or is it better understood as a connected community where cooperation is as important as competition? The wood wide web suggests the latter, revealing that trees are not isolated individuals, but members of an underground commune where support and communication flow through fungal intermediaries, linking the forest into something greater than the sum of its parts. One of the quietest but most important roles fungi play is in carbon storage. Through partnerships with plants, fungi help move carbon from the atmosphere into soil, where it can remain locked away for hundreds of years. In this way, fungi act as allies in stabilizing Earth's climate. The process begins with photosynthesis. Plants capture carbon dioxide from air and use sunlight to convert it into sugars and other organic compounds. Some of these carbon-rich compounds stay in the plant, building leaves, stems, and roots. But in plants with mycorrhizal partnerships, a significant portion flows underground to the fungi, payment for the nutrients and water the fungi provide. The fungi use some of this carbon for their own growth and metabolism, but they also transform it into compounds that can persist in soil for long periods. Fungal cell walls contain tough molecules that decompose slowly. Specialized compounds called glomalin, produced by certain mycorrhizal fungi, are particularly stable, remaining in soil for decades or centuries after the fungal hyphae that produced them have died. These compounds bind to soil particles, improving soil structure while locking carbon away from the atmosphere. When fungi decompose dead plant material, they release some carbon back to the atmosphere as carbon dioxide, but they also transform much of it into more stable forms. The partially decomposed organic matter becomes humus, the dark, carbon-rich component of healthy soil. Humus can persist for hundreds or thousands of years, representing a vast reservoir of carbon removed from the atmospheric cycle. Mycorrhizal networks enhance this carbon storage by extending deep into soil. While plant roots typically occupy the upper layers, fungal hyphae penetrate much deeper, reaching soil layers where carbon can be stored even more stably. Deep soil carbon is less susceptible to disturbance and decomposition, meaning it remains sequestered for longer periods. By moving carbon deep underground, fungi create long-term storage that helps balance the global carbon cycle. The scale of fungal carbon storage is substantial. Scientists estimate that mycorrhizal fungi alone may transfer several billion tons of carbon into soil each year worldwide. This represents a significant portion of the carbon fixed by terrestrial photosynthesis. Without fungi mediating this transfer and transforming carbon into stable forms, atmospheric carbon dioxide levels would be noticeably higher, with corresponding effects on global climate. Forest soils, particularly in old-growth forests with well-established fungal networks, store more carbon than the trees themselves contain. A mature forest's carbon budget includes not just the visible biomass, but the vast repository underground, much of it in forms created or stabilized by fungi. When forests are cleared and soils are disturbed, this stored carbon can be released back to the atmosphere, contributing to climate change. Protecting forests means protecting not just trees but the fungal networks and carbon stores they maintain. Agricultural systems with healthy fungal populations also store more carbon than those where fungi are suppressed by tillage or fungicides. Regenerative agriculture practices that support mycorrhizal fungi, such as minimizing tillage and maintaining diverse plant communities, can increase soil carbon storage. This offers a strategy for removing atmospheric carbon while simultaneously improving soil health and crop productivity. Climate scientists are increasingly interested in the role of fungi in carbon cycling. Models that predict future climate scenarios must account for how carbon moves between atmosphere, plants, and soil. Fungi are key players in these models, affecting both how much carbon gets stored and how long it remains sequestered. Understanding fungal contributions helps refine predictions and identify strategies for managing carbon more effectively. Research continues into how different fungal species and communities affect carbon storage. Some fungi are more efficient than others at creating stable carbon compounds. Some ecosystems, particularly in cooler climates where decomposition is slower, accumulate more fungal-derived carbon than tropical systems where rapid decomposition releases carbon more quickly. These variations matter for understanding global patterns and identifying where conservation or restoration efforts might have the greatest impact on carbon storage. These ideas can float through your mind gently, settling like spores on soil. There's nothing you need to remember or hold on to. The essential truth is simple. Fungi work quietly underground, taking carbon from air through their partnerships with plants and locking it away in soil. In this patient, invisible work, they help stabilize the climate that allows life to flourish, offering one more example of how these often overlooked organisms support the systems that sustain us all. In 1928, Nazi Alexander Fleming noticed that a mold had killed bacteria in his laboratory. This chance observation led to penicillin, the first widely used antibiotic which has saved countless millions of lives. Though not a mushroom, this mold belongs to the same fungal kingdom, showing how deeply fungi have shaped human history and medicine. Fleming, a Scottish bacteriologist, was studying Staphylococcus bacteria in his laboratory at St. Mary's Hospital in London. Upon returning from a vacation, he noticed that one of his bacterial culture plates had become contaminated with mold. Rather than simply discarding the ruined experiment, he observed it more closely and realized that bacteria near the mold had been killed. The clear zone surrounding the mold colony suggested it was producing something that destroyed or inhibited bacterial growth. The mold was identified as Penicillium notatum, a common species that grows on old bread and rotting fruit. Fleming named the antibacterial substance it produced penicillin. He published his findings, but initially struggled to purify and produce enough penicillin for medical use. The substance proved unstable and difficult to extract in sufficient quantities. For more than a decade, penicillin remained a laboratory curiosity with recognized potential but no practical application. The situation changed in the early 1940s when chemists Howard Florey and Ernst Chain developed methods to produce penicillin in larger quantities and greater purity. Their work, motivated by the medical needs of World War II, transformed penicillin from a laboratory oddity into a life-saving medicine. Soldiers who would have died from infected wounds survived thanks to penicillin treatment. The antibiotic prevented deaths from bacterial pneumonia, sepsis, and other infections that had been major killers throughout human history. The impact was immediate and profound. Before antibiotics, a simple bacterial infection could be a death sentence. Surgery was riskier because post-operative infections often proved fatal. Childbirth carried dangers from bacterial infections. Diseases like tuberculosis and strep throat killed millions. Penicillin changed all of this, making bacterial infections manageable conditions rather than potential death sentences. Life expectancy increased markedly as antibiotics became widely available. The discovery inspired searches for other antibiotics, many of which also came from fungi and bacteria. Streptomycin, tetracycline, and numerous other antibiotics were discovered in the following decades, each targeting different bacterial species or working through different mechanisms. The antibiotic era had begun, fundamentally transforming medicine and public health. The mechanism by which penicillin works is elegant. It interferes with bacterial cell wall synthesis, preventing bacteria from building the protective walls they need to survive. Without intact cell walls, bacteria burst from internal pressure and die. Human cells, lacking this type of cell wall, are unaffected by penicillin, allowing it to target bacteria specifically without harming the patient. This selectivity made penicillin remarkably safe and effective. Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine in 1945 for their work on penicillin. Fleming's Nobel lecture emphasized both the power of penicillin and the danger of misuse, warning that bacteria could develop resistance if antibiotics were used improperly. His warning proved prescient. Over the following decades, antibiotic resistance became an increasingly serious problem as bacteria evolved mechanisms to survive antibiotic treatment. Despite challenges with resistance, penicillin and its derivatives remain important medicines. Modern versions have been modified to work more effectively, last longer in the body, or target bacteria that have evolved resistance to original penicillin. The basic molecule discovered by Fleming continues to save lives nearly a century after its initial discovery. The story of penicillin reminds us that fungi are chemists of extraordinary sophistication. They produce antibiotics not for human benefit, but to fight their own battles against bacteria in soil and on decaying matter. Fungi and bacteria have competed for resources for hundreds of millions of years, driving the evolution of chemical weapons on both sides. Penicillin and other antibiotics are products of this ancient chemical warfare, compounds refined by evolution to be highly effective at killing or inhibiting bacteria. The lesson extends beyond penicillin. Fungi produce thousands of biologically active compounds, many of which remain unstudied. Some fight viruses, others suppress immune responses, still others show potential against cancer cells. The fungal kingdom represents a vast library of chemical compounds, each evolved for specific purposes, many of which might have medical applications we have not yet discovered. Every time we encounter a new fungal species, we potentially encounter new chemistry that could lead to new medicines. As this long, gentle journey through the fungal kingdom comes to a close, perhaps you can sense how these organisms weave through every aspect of life on Earth. From antibiotics that save human lives, to networks that connect forests, from spores floating in the stratosphere to mycelium breaking down stone into soil. Fungi are everywhere, working quietly, shaping the world in ways both vast and subtle. Let these thoughts rest lightly in your mind, settling without effort, as you drift toward whatever comes next, whether sleep or simply peaceful rest.